antibodies is discussed, and this is what it's all about. Initially, the B cell is recognizing microbes via IgM and IgD on the surface of cells. There can be others. Is this thing too loud? Is it louder than normal? Okay. There can be supportive signals. This is very important. This helper T cells and other stimulants. And then the B cells multiply, and then what they become is, in actual fact, antibody-producing factories, as I explained. And these then change their shape, and that's why the shape is changed here in these little diagrams. So these are activated B cells, and these are then plasma cells that make huge amounts of antibodies. And initially in the immune response, you find that there's IgM produced. Now watch here carefully. Look on the left-hand side here. It says IgM and IgD on the surface. And then what happens is, initially when the immune response starts, then it starts releasing them. They are also IgM, but now they're pentameric IgM. And there's a very specific reason for that, and we will discuss that shortly. Or, after a while, they can start producing IgG. And this story about isotype switching, different types of antibodies are called isotypes. And so it changed its isotype from IgM initially to IgG. And this IgG floats around the system, and it's very effective, and that's why it changes to IgG. I get to the reasons why it's better, but that's what's changed. And then I also explained to you that what happens is the actual shape of the antibody binding site changes. It improves. It improves so that it can fish out better rare antigens. So this is towards the end of the immune response, and it's trying to mop up and kill every last bug. So it changes its affinity, and then very importantly, in order to be able to overcome a next infection much more effectively, you get memory cells that are formed, right? And that I also explained to you that this formation of memory cells is the principle under which vaccination works. You get challenged with an antigen, you make a short immune response, and then you pack away memory cells. Next time around, you're much better off to be able to overcome these infections. So here on this scheme is shown to you a number of different types of things, and I'll just call them that B cells can recognize. And the first thing that B cells can recognize are protein antigens, and this is very important. They can also definitely recognize protein antigens, and T helper cells assist in this process. So there's going to be a whole spiel about how T helper cells can help T cells. But then there are funny things. Remember, in T cells, I told you, a presentation every time was MHC1 or MHC2, right? And all of our discussions were about protein antigens. But B cells can, funnily enough, make antibodies against other molecules as well. They can make antibodies against polysaccharides and lipids and things like this. And this sounds perhaps contraindicatory, but if you remember from microbiology, microbes make on their surfaces often polysaccharides or lipopolysaccharides. So it's very important for you to actually recognize those as being foreign structures. So it's important not only to recognize proteins, right? And that's the important principle here. Antibodies help us with mopping up other stuff as well. This dynamic of the immune response we've discussed, and we discussed this in the introductory section. The important thing is as follows. Low numbers of naive cells. If an antigen gets into your body, maybe 10, 20, slightly different shapes that can recognize an antigen. What then happens is the antigen binds to them. This causes them to become activated. They increase in number. Why do they increase in number? Because if they didn't increase in number, one cell can't make enough antibody molecules to be able to mop up the antigen. So that's why you always have this increase in number. They increase in number, they become plasma cells, they become antibody-producing factories. They release, first time around, they release mainly IgM and less IgG. Both of these are shown to you here, but more IgM almost than IgD. Then what happens is you overcome the infection. These antibodies lead to the killing of the antigen. So there's no point in making more antibodies. These plasma cells have done what they need to do. They burn out, they die, and the whole antibody production. However, you pack away memory cells, right? Second time around, antigen comes into the system. Instead of 10 or 15 or 20 cells, you've now got like 10,000. And you can kick off this process far faster. And you can kick it off with much greater numbers. So what happens is, this diagram, look at the curve here quickly. Here, initially, in the first immune response, you're looking at like 6 to 8 or 10 days. And here, very, very rapidly. You can see that within a few days, the antibody levels kick up. If this is, say, flu, first time around, you get like a sick, right? And second time around, you feel any symptoms, you just feel a sniffy one day, and then the next day you're on top of it, right? So this is what this immune memory is about. Again, multiple production of these cells, massive amounts of antibody, now second time around primarily IgG, and then control, right? This has cost you a huge amount of energy. These cells have multiplied, they've now become anti-producing factories, they've produced vast amounts of protein. That's why you feel pop when you're sick, right? But after that, you don't want to utilize that amount of energy, so you switch the whole process off, but of course you make memory cells again. So you've got enough memory cells to tide you over for next time around. As a matter of fact, what you find is that those memory cells go up. They will be better equipped for next time around. And this can carry on for years. I got exposed to avian influenza from birds in 2010, and in 2011 I went to a funny thing at which there were a whole lot of vets and people working on ostriches which came together. There was a lady that offered to draw blood from people. She drew a blood sample from me and discovered that I had the highest titer, that's how we talk about antibody amounts, of anyone in the whole audience against bird flu. So I'm very safe. Birds can't do anything to me. I've got huge amount of memory cells packed away against bird flu. I've got huge memory against bird flu. So I'm special. Okay. Here's the dynamic. This is just in tabular form. We don't need to look at this. What happens? This is a diagram which you have seen umpteen times before, right? This is the activation signal. The microbe binds to the antibody sits on the surface of the B lymphocyte. This can be IgG, at least IgM or IgD. When you see inside the cell, there's a whole signaling cascade, and what happens is you have a whole lot of transcription factors that get activated, and those transcription factors activate the genes that make the antibodies. Right? So all of a sudden now, this produces, these transcription factors switch on antibody production big time. Very easy to understand. I'm not going to ask you the details of the signal transduction cascade. You've seen all this before, protein kinase C and so on and so forth, RAS and PKC and ERK and JUNK and all these things. You know more about this than I do. The interesting thing is that you've done this before in this course, right? And you've also done transcription factors, right? So in actual fact, what you're seeing here is a combination of that, but now in some sort of a signaling system where you see what the effect is. So you've got signal transduction, plus you've got transcription factors and control of gene expression that's activated in order to produce antibodies. So in this phase here, this is signal transduction, and then there's massive activation of gene expression to produce these antibodies. Right? Now, this is a funny thing here, and this is one that almost runs ahead of time. And what this is telling us is that there is in actual fact not always one signal, but a second one as well. If you were to go back to the first, or the chapter on antibodies, then you would see that there's something about activation of B cells which says that it works when there are two signals. Right? If you can go back to that when you work through this, hopefully sometime before the test that you write. Go back and have a look. There's two signals. Now what you're seeing here is some other microbe. And this microbe is recognized now by the antibody receptor, right? The part of the antibody that can recognize it. But at the same time, this microbe's been floating around the system. And innate immunity activates complement via the alternative pathway. This is something that you heard a couple of lectures back, so it's rusty. But the bottom line is that that complement results, the innate immunity results in this C3e component being deposited on those bacteria. And what this does is this is the innate immune system which is already tagging this bacteria as being something that needs to be eliminated, right? And what is happening here is this is now recognized by a further receptor. So there's not only one receptor signal via the antibody, but a second one via complement. And just go back to those things and it says there's two signals, and this is all that this is about. This is a second way in which the immune response here is activated, in which B cells are activated, and it's actually innate immunity, and listen carefully, helping adaptive immunity. And this happens quite a number of times. So innate immunity is what you kick off with, what you try to get the thing under control with, and here adaptive immunity is kicking in, and the two things work together. This is what makes this quite complicated. Now, these are also these further basically indications here of what happens during B cell activation, and I'm going to go over this rapidly. Mitosis, they make additional receptors so that they can respond to other signals, and then they start with low-level secretion of antigens. So this is during contact with antigen. Now, these signals to let B cells multiply, these additional signals, often also come. Remember I said just familiar with that complement thing, with a second signal? Something else that can also give a second signal is a T helper receptor. And now, what happens here is like a funny hybridization of what I've told you about certain things before, right? And this funny thing is that if we look here now, what this scheme is showing you is somewhere inside the lymph node, right? And inside this lymph node, there are areas which are full of B cells, and then other areas where there are B cells and T cells, right? Here is a B cell, and this B cell is now confronted with an antigen. Here's this microbe or something that's coming in, and it's got a protein, and this protein has got a certain form that is complementary here to the B cell. So this is one of 10 million B cells that's got the right recognition site, and it can recognize the antigen. So it's recognizing it via an antibody molecule. And now, careful, because this can be confusing. What it does is it phagocytoses that bug, right? It phagocytoses that protein. This is the same as though it were a macrophage. Can you remember we've discussed macrophages that gobble stuff and then present things on the MHC, right? So the B cell here is almost serving as though it were a macrophage. It phagocytoses the antigen and presents the antigen via MHC2. And it's via MHC2, very important, right? So phagocytosis of the organism and presentation of MHC2. Then somehow or other it migrates out here. Don't worry about all this migration, there's far too much detail here. But importantly now, because it's got an antigen which it's presenting via MHC2, this can now be recognized by a T cell that's got a suitable receptor to recognize that antigen. You've seen this before, right? You've seen this where I've told you that macrophages can be, that can present an antigen, and then that the T helper cell can help it, right? This is very similar to that picture. Basically what is happening is that the T helper cell can recognize this and can activate the B cells, can help the B cells to be activated. And what this means is that those B cells can become plasma cells, and they can now go into massive antibody production. So, second signal in this instance comes from the T helper cell, right? So this is just one of those things that you've got to be careful with, and we come back to this time and time again. This is what happens. So the antigen gets recognized, gets phagocytosed, gets presented on MAC2, and the CD4 positive T helper cell can recognize it. Okay? So this is a complicated thing, but basically you've seen this before, and in this way the B cell can signal that it needs help. This is in the case of a protein antigen. This is too much detail, I'm going to skip this, but now, once the B cell has become activated, what it must do is, it must now starts off by making IgM.